The cell membrane, also known as the plasma membrane, is a dynamic and intricate structure that forms the boundary of every living cell. It's not just a passive barrier, but an active interface that governs the interactions between the cell.
Imagine a mosaic that is constantly shifting and rearranging its pieces. That's a good way to visualize the cell membrane. Its primary components include lipids, approximately 40 to 50% by weight. The workhorses here are phospholipids, which form a double layer called the lipid bilayer. Each phospholipid molecule has a hydrophilic or water-loving head and two hydrophobic or water-fearing tails. In the bilayer, the hydrophilic heads face the watery environments inside and outside the cell, while the hydrophobic tails cluster together in the interior, creating a barrier to water-soluble substances. Cholesterol, another lipid, is interspersed within the phospholipid bilayer in animal cells. It helps to regulate membrane fluidity, making it more stable and less permeable to small water-soluble molecules. Glycolipids, lipids with attached carbohydrate chains, are found on the outer surface of the cell membrane. They play a role in cell recognition and signaling. Proteins, approximately 50 to 60% by weight. These are the functional components of the membrane, carrying out a wide array of tasks. They can be broadly classified into two types based on their association with the lipid bilayer. Integral proteins are embedded within the hydrophobic core of the lipid bilayer. Many of these span the entire membrane and are called transmembrane proteins. They often function as channels, transporters, or receptors. Some integral proteins are only partially embedded and may be anchored to one side of the membrane via lipid tails. Peripheral proteins are not embedded in the lipid bilayer but are associated with the membrane surface through interactions with integral proteins, or the polar head groups of phospholipids. Glycoproteins are proteins with attached carbohydrate chains also found on the outer surface of the cell membrane. Similar to glycolipids, they are involved in cell recognition and interactions. Carbohydrates, approximately 1 to 10% by weight. These are usually attached to lipids, forming glycolipids or proteins, forming glycoproteins on the extracellular side of the membrane. This carbohydrate layer is called the glycocalyx and is crucial for cell-cell recognition, adhesion, and protection. Water and ions. Water molecules are associated with the hydrophilic heads of phospholipids, and ions can be found adsorbed to the membrane surface due to the negative charge of some phospholipid head groups. Water also fills the pores of some channel proteins.
Functions of the cell membrane. The cell membrane performs a multitude of essential functions that are vital for the cell's survival and interaction with its surroundings. The primary role of the cell membrane is to act as a selective barrier, controlling which substances can enter and exit the cell. This semi-permeability is crucial for maintaining the cell's internal environment, allowing essential nutrients in while keeping harmful substances out, and facilitating the removal of waste products. Transport of molecules. The membrane facilitates the transport of various molecules across it through different mechanisms. Passive transport, for example, simple diffusion and facilitated diffusion, does not require energy and relies on the concentration gradient of the substance. On the other hand, active transport requires energy, usually in the form of ATP, to move substances against their concentration gradient. Membrane proteins like pumps and transporters mediate both facilitated and active transport. The cell membrane contains numerous receptors that bind to signaling molecules like hormones, neurotransmitters, and growth factors in the extracellular environment. This binding triggers intracellular signaling pathways, allowing the cell to respond to external cues. Cell adhesion and interaction. Membrane proteins and carbohydrates are involved in cell-cell recognition and adhesion, allowing cells to interact and form tissues. Specialized junctions between cells, mediated by membrane proteins, facilitate communication and maintain tissue integrity. Maintaining cell shape and structure. The cell membrane provides structural support to the cell and helps maintain its shape, often in conjunction with the cytoskeleton, a network of protein fibers within the cytoplasm that can anchor to membrane proteins. Enzymatic activity. Some membrane proteins act as enzymes, catalyzing reactions at the cell surface. Endocytosis and exocytosis. These are processes by which the cell can engulf large particles or secrete substances, respectively, by altering the shape of the cell membrane and forming vesicles. Ion channels are a specific type of integral membrane protein that form pores or channels across the lipid bilayer. These channels are highly selective for specific inorganic ions such as sodium, potassium, calcium, and chloride, allowing them to pass through the membrane down their electrochemical gradient, which is a combination of concentration gradient and electrical potential difference. Here's a closer look at ion channels. Ion channels are typically formed by several protein subunits that assemble to create a water-filled pore spanning the membrane. The narrowest part of the pore, called the selectivity filter, determines which ions can pass through based on their size and charge. The opening and closing of ion channels, known as gating, is tightly regulated. Different types of stimuli can control gating voltage. Gated channels open or close in response to changes in the electrical potential across the cell membrane. These are crucial for generating and propagating electrical signals in nerve and muscle cells. Ligand-gated channels open or close when a specific molecule, the ligand, such as a neurotransmitter, binds to a site on the channel protein. These are essential for synaptic transmission and other forms of chemical signaling. Mechanically gated channels open or close in response to physical forces or mechanical stimuli, such as touch or pressure. Other gating mechanisms include temperature changes, light, and intracellular signaling molecules. Ion channels play critical roles in a wide range of physiological processes, including nerve impulse transmission. Voltage-gated sodium and potassium channels are essential for the generation and propagation of action potentials in neurons. Muscle contraction. Voltage-gated calcium channels trigger muscle contraction by allowing calcium ions to enter muscle cells. Regulation of heart rhythm. Ion channels control the electrical activity of the heart. Secretion ion channels are involved in the release of hormones and neurotransmitters. Sensory transduction. Mechanically gated channels in sensory receptors convert physical stimuli into electrical signals, maintaining cell volume and osmotic balance. Ion channels help regulate the movement of ions and water across the